Hello, I'm Haritha. I'm a chemistry PhD student from Dr. Dong's group. Today, I'll be talking about my project on the synthesis of pH responsive polymer peptide hydrogels for antibacterial therapy. So, to begin with, I would like to introduce you to some background research that inspired us to undertake this project. So, recently, our group has demonstrated the synthesis of pH responsive self assembling multi-domain peptides, which forms a sandwich like supramolecular structure, so as shown here. And when we introduce a tertiary amine containing no natural amino acid, we observe the disassembly of the nanofiber. So essentially, they form monomers at acidic pH and nanofibers at neutral to basic pH. And then we decided to use these peptides for antibacterial therapy, where we found that after disassembly under acidic condition, these peptides show an enhanced binding towards bacteria, and these peptides also exhibit good cytocompatibility. So we also found that these peptides at higher concentration can form hydrogels. So hydrogels are basically cross-linked peptide nanofiber networks, and they're capable of retaining a large amount of water, and they can also maintain 3D hierarchical structure. So you can see an image of hydrogel over here. And peptides hydrogels are used for various applications such as wound healing, 3D bioprinting, drug delivery, etc. So therefore, using this pH responsive peptides for the formation of hydrogel will be an additional advantage for target specific therapy, where we can use pH as a stimuli. But the challenge we had here is the strength of the hydrogel. So these peptides form very weak hydrogels, and they're not good enough for the biological applications. So therefore, our aim for the study is to increase the strength of the hydrogel by incorporating more covalent bonds that can interconnect the nanofiber. So for this study, we'll be using a thiol malamide click chemistry. And to connect a polymer, which is 4-arm peg malamide. So this polymer has 4 reactive malamide groups, and we'll be reacting this polymer with a self-assembling peptide, which contain a thiol group. So once we have the peptide polymer conjugate, we will introduce more peptide uh, so that we can also use the hydrogel forming property of the self-assembling peptides. And then we will encapsulate the drug, and then we'll study the pH dependent release. So now I'll show you some of our experimental results. So far, we are able to synthesize peptide polymer conjugate, which was confirmed by mass spec using MALDI. We were also able to separate peptide polymer conjugates using HPLC at a good yield. And then when we used co-assembly method of peptide and peptide polymer conjugate, we observed a strong hydrogel formation. So these are some of the images of the hydrogel formed. Now for the future work, we'll be doing rheological studies first to measure the strength of the hydrogel, and then we'll perform antibacterial test uh, at different pH. And then for the release study, our plan is to encapsulate the gel with an anti-diabetic drug as well as glucose oxidase enzyme. So glucose oxidase is an enzyme which convert glucose into gluconic acid. And so we are planning to use high glucose concentration in diabetic patients to generate an acidic environment by the formation of gluconic acid. This will help in the pH dependent release of the drug. And finally, we'll be doing some animal experiments and using diabetic mice models for the treatment of periodontitis diabetic comorbidity. So, in conclusion, we have demonstrated the use of malamide thiol chemistry for the synthesis of peptide polymer conjugates, and the co-assembly of peptide and peptide polymer conjugate has been implemented to develop a stronger hydrogel. And the results from this study will provide valuable insight into the fundamental understanding of double network peptide polymer hydrogel formation, which has a very important potential for bacterial cell targeted therapeutic delivery as well as drug delivery application. Thank you for listening.